Motor nerves do not simply transmit electrical signals to muscles, they also maintain the health and structural integrity of the muscles they supply through ongoing trophic influences. The word trophic here means nourishing, referring to the continuous chemical and molecular signals that nerves provide to keep muscle tissue organized and functional. When a muscle is denervated, meaning its nerve supply is cut off, the consequences are swift and severe. The muscle undergoes rapid atrophy, losing more than half of its original bulk within just 2 to 3 months. This is not a gradual decline, it is a dramatic, fast loss of mass happening within weeks of denervation. Beyond mass loss, the structural organization of the neuromuscular junction itself begins to break down. Under normal conditions, nerves actively cluster acetylcholine receptors to the end plate region, concentrating them precisely where transmission must occur. When the nerve is lost, this clustering signal disappears. Receptors fail to stay concentrated and instead spread out diffusely across the entire muscle membrane surface, rather than remaining confined to the end plate zone. This receptor spreading is the structural basis for a phenomenon called denervation hypersensitivity, where the entire muscle surface becomes responsive to acetylcholine or acetylcholine-like drugs. In an innervated muscle, only the end plate zone is sensitive. In a denervated muscle, every part of the membrane reacts. This carries a dangerous pharmacological implication. Administering succinylcholine, a depolarizing neuromuscular blocking agent, to a patient with denervated muscles can trigger simultaneous channel opening across the entire muscle surface, driving massive efflux of potassium from inside the muscle cells into the bloodstream, and potentially causing life-threatening hyperkalemia. Motor neurons provide trophic factors including calcitonin gene-related peptide CGRP, and nerve-derived growth factors such as agrin and neuregulin, which support NMJ maintenance, receptor clustering, and mature receptor stabilization. Nerve-induced electrical activity suppresses extrajunctional AChR expression. After denervation or severe inactivity, immature fetal-type receptors begin to be synthesized within hours, and over several days can spread over the whole muscle membrane. Denervation causes muscle atrophy despite adequate nutrition, and without reinnervation, there is progressive atrophy and eventual muscle destruction. Muscle fiber type composition also shifts over time. Denervation is suggested by weakness with lower motor neuron signs such as hypotonia, depressed reflexes, fasciculations, and atrophy after weeks. On needle electromyography, EMG, fibrillation potentials and positive sharp waves indicate denervation, while biopsy may show angular atrophic fibers and later fiber type grouping from denervation-reinnervation cycles. Next video, we will discuss fasciculations and fibrillations.